This is the last review video for Unit 1. This is the part of the immune system we cover in Unit 1. And that is the big themes in immunity, the differences between innate and adaptive immunity, and then parts of the innate immune system. So the big themes include things like tolerance of self. What does that mean? It includes immune memory. What does that mean? We also talked about receptors and ligands, because that shows up all the time. Another thing that you need to be able to do is tell me the differences between innate and adaptive immunity. And you've probably done this in other courses, so I want you to pause the video and write a lot of different differences between innate and adaptive immunity, and then I will show you our list in this class. See how close you get. So this is the list we use in this class. We can compare how evolutionarily ancient they are. We can talk about their essential qualities. The innate system is fully defined in our DNA, in every cell in our body, from birth. And it's the same in everyone. Whereas the adaptive immune system changes, the DNA even changes. And so the adaptive immune system gets better and better and better as we get older and older. But it is slow compared to the fast responses in the innate immune system, and then other things. So go back and watch the video for that, or go back and look at the slides or the study guide or whatever works for you. Okay, so what were the parts of the innate immune system? We started with physical parts that mostly came down to skin and sloughage of skin, where the cells fall off and carrying with them any dangerous thing that landed on us. Respiratory mucus and other mucus, but respiratory mucus is amazing because it gets pushed up out of the respiratory system and sent down into the stomach, where anything that tried to get to our lungs gets killed. And there are other physical parts of the immune system, but those are the big ones I like to think about. And then the chemical parts of the innate immune system include the antimicrobial molecules like defensins, lysozyme, transferrin, antiviral signaling molecules like interferons, sweat. I can never decide. Should it be chemical? Should it be physical? Because when it dries, it turns the skin into a desert. I really can't decide whether that's physical or chemical, and it really doesn't matter. Just know sweat protects us by driving down the water activity of skin. So skin is something like this in terms of water activity. And so bacteria like Staphylococcus are able to grow on skin. None of these bacteria can grow on skin. And that's cool. And then phagocytosis was the first big thing we looked at as a cellular part of the innate immune system, because this is what the cells do. This is what the anti-microbe cells do. Cells that are going to protect us from bacteria and viruses are generally going to do phagocytosis. So we looked at what phagocytosis is and why cells do it. And then we looked at the cells that do phagocytosis. So quick, pause the video and list cells that can do phagocytosis. So hopefully you listed macrophages, dendritic cells, and neutrophils, and maybe Kupffer cells and endothelial cells. And then for each one, can it do antigen presentation? Can it clean tissues? Can it modulate inflammation? Those are things to know about all these cells. And then, really, how do cells know what to eat? And that comes down to pattern recognition, where cells of the innate immune system look for PAMPs using their PRRs. So go back and look at what a PAMP is and what a PRR is, because those are things you need to know. And so PRRs are pattern recognition receptors. PAMPs are pathogen-associated molecular patterns. So what are some examples of PAMPs? Pause the video and tell me. When in doubt, go back and look at your diagrams of bacterial cells, because what you're drawing there in a lot of cases are PAMPs. Things like peptidoglycan. Why is that a PAMP? Why is that a pathogen-associated molecular pattern? Well, where do we see it? Where would a macrophage encounter peptidoglycan? Never, unless there's a bacterial infection. Where would a macrophage encounter lipopolysaccharide? Never, unless there's a gram-negative bacterium in the wrong part of the body. What about teichoic acids? What about double-stranded RNA? We haven't talked about double-stranded RNA. That's Unit 2. But these are examples of PAMPs, and you should know that. So PAMPs are going to be any molecule a pathogen would have that a self cell would never have. And what else? That's pattern recognition. Pattern recognition is cool. Opsonization. Self versus not self. Lots of good stuff. And then the last part of this, the last thing from Unit 1 to know is inflammation and fever. So inflammation and fever, there are a few slides about it, just saying what they accomplish, what they are. So make sure you go back and look at inflammation and fever. Whatever you learned about them in A&P is probably adequate for our purposes. So that's it for the parts of the immune system from Unit 1. That's it for the Unit 1 review. Good luck. Post questions to the Q&A, and expect to do great on the upcoming exam. And I'll see you there.